thank you very much, everybody, for attending this webinar on colour, I'm going to say, in inverted commas, because it's more than that, with Professor Silvia Vignolini. I'm Louise from Cambridge Network, which is a business based in Cambridge, UK, and we aim to facilitate cooperation, training and recruitment for our members, who are from about 1,200 organisations in and around the high-tech centre of Cambridge. This talk is one of a series of termly talks from the University of Cambridge's Maxwell Centre, and I'll hand over to Monica shortly, who can tell you more about that and introduce to our speaker. Firstly, I've got the boring housekeeping bit to do. We love your questions, so please ask them in the chat box below. We'll ask you to keep on mute just to keep the background noise down, really, so we can all hear what's being said. I'll keep an eye on that. Monica and I will keep an eye on that. We have a 1-hour session and we are recording it as well, so you will be able to catch up with it afterwards if you miss anything. I think that's everything I've got to say. So I will hand over to you, Monica, and to do the rest.
Thank you very much, Louise. My name is Monica Lucena. I work at the Maxwell Centre as the engagement coordinator, and every term we organise a talk together with Cambridge Network where we try to bring what's exciting in our research community, and that could be of use to commercialization or to people who may be interested in that particular field. With regards to our speaker today, Silvia is a truly multidisciplinary researcher, and she's a leading expert on light propagation in complex photonic structures. She's a physicist by training and is a professor in the Department of Chemistry. Silvia's group uses optics to understand the assembly of naturally occurring photonic structures and applies that knowledge to fabricate novel biomimetic sustainable materials. Is that a good summary, Silvia?
Yeah, I think it's good. It's good.
So I'll pass on to you.
Thank you for inviting me, and then thank you for everyone attending this talk. I really have only have one screen, so I don't have a double screen, so I don't have an idea to whom I'm talking to. I'm just talking to the slide. So I hope that I would be able to entertain you for the next half an hour, 40 minutes with what we do. So we will talk, as they say, we will talk about color. And I would like to just remind to people something that we often give it for granted, that color actually it's an important part of our communication. So it's something that we need in our life, and we use it on a daily basis, and that influences our choices, and not by chance I put this picture of this supermarket with all these multiple differently colored packages, because it's a way that we are trained by evolution to choose, right? So the appearance of an object attracts us or not, and therefore the possibility to manipulate coloration, it's important, it's really important in human communication. But not only for human, but in any aspect of any other animal living on this planet. And this is essentially, in fact, what we are interested in. We try to understand how living organisms produce this color, not only with pigment, I will show you in a moment, but with the complex structure. And the idea is to understand this, to understand how it's possible to manipulate material to produce coloration in nature, is to translate this principle from nature to application, and then try to produce material that are more sustainable, but that still provide the same range of coloration that we actually need in our daily life. So these are the two aspects that we generally do. So at the beginning, usually when I give a talk, I also talk a bit more about what we talk about in what we observe in nature. But this time I will mainly focus the attention on how we can make this sustainable biomaterial. So before I start, I think I really need to essentially thank the people that have done this fantastic work, and especially the part on the cellulose self-assembly that will be the main part of the work has been led in my group by Bruno Facapetesic and Richard Parker, that are the two main postdoc working on this topic, and also I think to acknowledge many other former members that essentially contributed to the work. Okay, so why do I say that we want to understand how nature make coloration to make more sustainable material? Because the way that we make colors and pigment today is not truly sustainable, I would say. In fact, we have to deal with pigments that are often obtained by chemistry methods that are far away from the green chemistry rules. And often when you hear about like pigments that are natural pigments that are plants-based, in terms of sustainability, you also need to consider the fact that you need to grow plants in order to produce this pigment, and the same growing, it will consume water and resources that actually could be like employed to produce crop. So essentially, there is always a fine balance when we talk about sustainability, and many aspects need to be taken into account. But if we talk about pigments and what is the pigment market, is one of the largest market in the chemical sector, and therefore it's a really important part of our industry. So we need to find solution on how to improve this for the future. And in fact, the majority of color that we see around is produced by what we call traditional pigment. Let me give you a better understanding. A traditional pigment is a molecule, so it's a chemical characteristic of the material, and you can synthesize it or extract it from natural resources. And what is happening is that when you illuminate this material, part of the light is absorbed and the absorption spectrum depends on the chemical composition of the material itself. And what is not absorbed is reflected back, scattered back, and this is what you perceive as a coloration in your eye. So this is what you see as a color. But actually, it's also possible to make color out of transparent material. And this phenomenon is different, it's called interference. And in fact, in this case, the light interacts with the material needs to be structured on the nanoscale, so that the color now that is reflected depends on the interaction of the material and the light itself. So what I think it's important is like the take-home message of this is really thinking about the soap bubble. The coloration doesn't depend on the material itself. You start with something that is transparent, like a water and soap solution. But then as soon as you structure it, you can obtain a color. And then you actually, depending on how you structure, you can obtain different coloration. And this interference mechanism, it's a characteristic of the light. So light is a wave. So you can imagine that the light of specific color have specific characteristic. When it interacts with a medium, you can, for example, create what you call constructive interference. So you can reflect a coloration, and light of other different coloration will not satisfy the same condition, so you cannot really reflect. So this interference mechanism is such that by changing the characteristic of the medium, you can really change in terms of structure, dimension, and size. You can really change the color that you obtain. So in principle, you can start from any material. And this is the concept of what you call structural colored pigments, yes? So this generally in industry, when you see like a metallized car that have this coloration that it's a bit metallic, that's how you say metallized, that it changes a bit depending on the angle of incidence. These are coloration that function in exactly this way, using this concept of interference. And also many glitter and cosmetic powder that we use that have this metallic appearance are essentially obtained by platelet material, that is where you have a layer of different materials that are arranged on top of each other in a periodic way, producing this interference effect. So they are also called interference pigment, in fact. So the mechanism is completely different from what we have on absorption. So, however, often to produce this pigment, or also you exploit this interference pigment, or also to boost the interaction with absorbing pigment, you combine inorganic material like titanium dioxide that are creating actually lots of problems. Not only for the environment, but also for the health. In fact, in some nation that there's already been this type of pigment paste of titanium dioxide, they have been banned. So there is at the same time, you are really needing to try to make new coloration and new color, but then you also need to take into account this, what are the right material to use. And I think that the solution can come really from nature, and that's why we called we like the idea to look at natural structure, because the most bright coloration that you obtain in nature are obtained by biomaterials, materials that are like chitin in case of this in these beetles, or even collagen. So this blue coloration of this baboon is the same collagen molecule that we have inside our skin, that we know that it's important to maintain the elasticity. It can also be exploited for color, or other simple polysaccharides like cellulose. So it is interesting to see and learn that actually in nature, the same variety of coloration doesn't necessarily rely on producing pigment, but it can be used, they can use the structural color, but they can make it out of material that are abundant and also sustainable. So what is important is that in nature, you don't find often this type of, you find also structure like this, but you can also find what we call the hierarchical structure. So you don't only have platelets where the material are arranged, but you have more into cellular type of shape. So you have a concept where the color is wrapped around the cell, because that's how this material are produced, this nanostructured material are produced within a specific cell. So the idea is that you're really playing with this concept of interference in hierarchical structure. So where you control multiple length scale at the same time, you can really obtain this really vast and rich appearance of color that we observe in nature. And I'll give you an example, because this is our main source of inspiration, and they are the helicoidal structure in the cell wall. So this is like a fruit, which is called Margaritaria. And interestingly enough, the skin of these fruits, like it's the skin of an apple, right? In this case, is colored. This really has this strong blue, vivid coloration, not because it contains a blue pigment, but because of how the cellulose, it's organized in the cell wall. So if you take a cross-section with the scanning electron microscope, and you look at the skin of the fruits, you can see that the cell, as I was telling you, have this hierarchical architecture. So they have a cell wall, and when you look inside the cell wall, you can recognize this tiny layered structure. And essentially, this layered structure made of cellulose that is comparable on a scale that is comparable with the wavelength of the light, essentially is what is responsible of creating this structural color. And here you can actually see it better, like this type of architecture is common not only in these fruits that I was showing you before, but it's common in many other different plants, by both other fruits, but also ferns. And nicely enough, it's essentially created only using one single material, cellulose. So why am I so keen with say, like, can we make color exploiting cellulose in the same way as plants can do? Like, and then not only copy the architecture, but also using exactly the same building block. Yeah. So this is like the challenge that we want to do. And the reason why we like to use cellulose in specific is because cellulose is a really good material in terms of human use. It has been used by our society since the beginning of the society. You think about pergaments and paper, right? It's a textile. These are like the material that are most have been used since ages. And in fact, the cellulose market is it's a really large market in terms of material produced. So even our glasses, the structure of our glasses, essentially it's a cellulose derivative that is a cellulose acetate. So this is we use it so much because it's abundant and renewable. In fact, it cannot only comes from wood pulp. So it's not that you have to cut forest in order to get cellulose, but you can get it from any plant's resources. And since it is in any plant resources, it's also edible and biocompatible. In fact, when you think about having a diet that is rich in fibers, these fibers essentially are cellulose fibers. In fact, cellulose comes as a fiber in nature. It comes as a natural fiber that it's what is provide the strength to the plant cell wall. Plants don't have a skeleton like us. So in order to be able to sustain the weight, and you think about a tree has several tons, can weigh several tons, in order to maintain this mechanical and rigid structure, you have skeletal polysaccharide, like in fact cellulose, that are creating fibrils. And these fibrils essentially are like themselves a really complex hierarchical architecture where you start with really simple glucose molecules. So these are like sugar molecules, yeah? And then they are linked together, but the way they are produced in the plant cell wall, they are like spinned out, this single molecular chain, and then they start to interact, forming a strong hydrogen bonding and create fibrillar, nanofibrillar structure that can be also highly crystalline. And that's what gives the mechanical support. And we actually use this crystalline part of the cellulose in order to make color. So obviously, I don't expect you to understand the details, because I'm just giving you an overview, but I hope that you will, I hope you believe me of what I say. So if you extract the cellulose nanocrystal from any plant's resources, from cotton, from wood pulp, and all the results that I'm showing you essentially are obtained mainly by cotton, that we take the cotton from the cotton linters. So essentially, you can even use the part of the cotton that is a leftover from the paper manufacturing company or for yarns, because they are too short to be exploited to make proper and resistive fibers. So we take this small leftover from this industry, and then you can do acid hydrolysis, and this acid hydrolysis mechanism release what we call cellulose nanocrystal particle, that if you look at them under the TM microscope, they look like this. So I don't have a scale bar, sorry, but they are about a few hundreds nanometer in length and only 3 nanometer in thickness. And magically, if they are in the right condition, when you put them in a water suspension, so simply having them in water, they start to interact. As you increase the concentration, they start to form what we call liquid crystal cholesteric phase. So you might have heard about liquid crystal display, and you know that somehow they have to do with color. This is an analogous from a natural point of view of liquid crystalline molecule. Instead of having molecule, now we have colloidal particle. So when we evaporate water, we increase the concentration of this cholesteric structure, and what is happening is that before they are dispersed in water, so they don't interact, and their distribution is random inside the suspension. But then as soon as you reduce the concentration, they start to feel each other, so they start to interact, and what they do, they start to form this cholesteric structure. And if you remember the few slides before, they create this layered architecture that it's really similar to what I've shown you in the case of the fruits. And in fact, you can get colored film. Let me show you a little bit with a higher magnification. If you look again in cross-section of the film that I just showed you, you can recognize this layered structure where you can really see the cellulose crystal, like in the case of the plant cell wall, on the same length scale that provides this blue coloration. So essentially, you are able to make a color out of the same material that you have in your paper. Yeah, this is like essentially the concept behind our work. So once you know how to control it, and once you know how to properly manage the self-assembly properties, you can really change the color in the entire visible spectrum. You can even reach in the ultraviolet and the infrared region. And you can really play also with the self-assembly. So by changing the composition, for adding other dopant inside the film. So this is just this colored here, this differently colored film are just obtained starting from exactly the same material, cellulose, and then we add some salt. So the only things that we do, we add really a little bit of salt, millimolar amount, so that the interaction within this crystal, this cellulose nanocrystals that we call CNC changed, and then we can tune this coloration. But then, and in this case, we obtain film. But then if you want to change also, for example, the mechanical properties of this film to make them more flexible or more bendable or more elastic, then you can simply need to add additional material, polymer like PEG. Here in this case, we just show the presence of a surfactant can completely change the mechanical properties. But many people have shown and demonstrated that using many other different type of polymer, you can completely change the mechanical response of the film, but at the same time maintain this nice coloration. What you can do, you can even change, when I say you can change the color, but you can also create gradient of color, because you can orient this cholesteric architecture in different direction. And when you can do that, let me just show you this movie here. The nice things of the structural color is that you can really, the color depends on the directionality that you are looking at it, and you can exploit this interference effect to create what we like to call holograms. So this is like, these are like flat films where the cellulose is essentially, the film is perfectly flat, but since the orientation of the, in this case of this photonic structure is in different direction depending on the angle, you obtain this really nice three-dimensional effect. And this is another video that hopefully will show, yes, where you can actually see that the color, you have this three-dimensional appearance, while in reality it's a flat 2D film. And this comes like because the light is reflected from this type of structure, it's going in different direction, giving the perception of three-dimensionality. So these are like just small example on what you can actually do starting only from simple cellulose. So you can really have different type of structure. So, and once you can create them, you can really fabricate it on large scale. The idea is that you can exploit them as proper pigments. So you can then cut them, grind them from film and then obtain coloration. But one thing I want to a little bit insist, because I was showing you the property of how the architecture matters in how you obtain the structure. I want to show you, like using the cellulose nanocrystal, how the optical properties of this material and the visual response that you obtain can change in function of this, what we call hierarchy. So we call hierarchy the additional structure on the micron scale that we add to this material. So I'll show you two examples, the one in having droplets, so we're having spherical type of structure, to the one that having like droplets that instead are evaporated on a glass slide. So this is what would you be more similar to the case of a pigment, more similar to also what you have in the plants, because you remember the cell, that the cell architecture is kind of spherical. And this is something that is more similar to when you, for example, print and you create a film. So in fact, we actually have used a printer. This is a printing method. And by changing the same way that we can make film of different color, you can just simply change the amount of salt in the suspension. And what you can do, you can change the coloration that you print from, let me just take again this laser pointer. You can essentially go from red to green to blue. And what is nice is that when you print this type of structure, since the layering, it's, as I was telling you, the structural color depends on the interaction within the light and the material. So if you change the directionality, you are changing the direction in which you are looking at the material. So if you instead of looking from the top, you look at a more angle, the same film, the same printed macrofilm will change color. So this is an effect which is called iridescent. And this is actually what you see when you look at this metallized car. Yes, so this platelet that you have in metallized car, essentially they look metallized, essentially because they have this type of effect, this iridescent, so that the colors slightly change depending on the angle of incidence. And then the way that it works, if you have a red structure, you tilt it, it looks kind of yellowish green. When you look green, it goes into the blue, and then when you do it blue, it shifts into the ultraviolet, and then we are not able to perceive it with our eyes. But it will reflect in the UV region. And this printing technique are pretty cool because you can actually then transfer it, you can print it on different type of substrates. And what I think it's actually very nice is that since you have this structure which is chiral, I didn't go too much into details, but you can also use it as a security feature. In fact, they have a specific, not only they interact with light, but they also interact with polarization of light. So if you wear glasses like the one that you have at the cinema, so polarization glasses, you can see that the color appear or disappear depending on which type of lenses that you are using. So what is nice is that, you know, this type of technology is fully compatible with inkjet type of printing. And this is like a work that we have done with an inkjet company that are producing printer, where we can actually showcase that our material can be printed on scale. And here I think it's really beautiful because we're having with two nozzles, you can essentially tune the coloration by simply adding a little bit of salt in any of these different suspension. So this is things that can be really have the true potential for scale up. So instead, again, if we go, I will show you the second example of the emulsified droplets, where you can see that how the hierarchy plays in terms of optical response. So, and I was telling you that when, like, this is what you usually observe in nature, that in plants, this kind of helicoidal architecture is always wrapped around the cell wall, so into like more or less spherical, or in this case, more ellipsoidal type of shape. So the idea is like, can we also replicate this hierarchy so to have a more rich optical response? And the answer is obviously yes. So here we use microfluidics, but in principle, you can even use other emulsifying technique. And we are working on the scale up of this technology. And now, instead of the self-assembly happening in a standard field, so while the water is evaporating in film in air, here you are evaporating water through an oil phase. And what is nice is that you can obtain particle, and we can change the size of this particle, that have the same type of layered structure that is what is responsible of the structural color. And in fact, you can obtain pigment particle that really now resemble the cell of these fruits, again in different coloration, going from blue to green to red. And what I was telling you, why the hierarchy is important, and this is something that I think it's really cool when you want to make pigment, is that if you have this type of architecture that are more spherical, when you embed them inside a coating, the optical response, it becomes much more angular independent, meaning that if you look them from any direction and you illuminate them from any direction, the color response remains more or less always green. Instead, if you remember the previous slide, when you print them and you have a really well-defined layer, like similar to the platelets, you can get a color that changes from green to red. So I think this is another really important take-home message that I would like you to have, because often when you hear talking about structural color and also in nature, you always think about always having a color that it's metallic, that change, and iridescent, that change in function of the angle. But in reality, in nature, and now also I show you here in this type of application, if you copy the hierarchy that natural structure offers, you can even brought this forward and actually get something that is possible to completely manipulate the optical response, not only in terms of color reflected, so green, blue, or red, but also in terms of color appearance, so how the color change in function of the angle. So like in this case, we can get angular independent color. And I like to stress that like lots of this material that we use are edible. So to convince you, these are some pictures of me when we were still allowed to have party. And this was like a party that we had during Christmas party of the group in the lab, where we use one of this material to color our cake and our food. And obviously it's always, since the structural color, it's really vivid when you have a dark contrast in between. It always really goes nice with the chocolate and chocolate-based cake. So this is a big advantage, a big plus. Independently from the nice coloration, however, the taste, I would say, is not the best. And in fact, I would say that it's not surprising that it doesn't taste anything, or it tastes a little bit like paper, because it's more or less the same material. But when you start, and you can think, yes, you can do it on small scale, but actually this same material that I was showing you before that we are eating, that is actually food grade. And by the way, we don't eat every our sample. We have special type of cellulose that we buy at food grade and never touches the lab, because you shouldn't be eating your own sample if they pass through a chemistry department. So this is important to remember. But if you do this properly, so if you use the food grade material and if you mix it properly, it's really easy to make this type of color, because in this case of this hydroxypropyl cellulose that is used in industry as a actually food thickener for making better foam, or for dough for people that have intolerances to gluten, essentially it's also like a material that has this really fantastic optical property. And since it's so abundant and easy to process, we were able to actually use on large quantity to make roll that are essentially made with this roll-to-roll machine, and they are highly colored. And what is nice is that, and these are the two persons that work on this paper, is that if you use them, and they are still in presence of water, you can actually exploit them as truly sensor material. So these are like, this is like a fully edible cellulose derivatives, where you can make pressure devices. So you can walk on top of this film, and then you have a direct colorimetric response that once you calibrate it into a value of a specific pressure, you can exploit it to make 2D map of pressure. And it's actually, the big advantage is that at the end, the material that is inside, it's so biocompatible, edible, and it's a cellulose derivatives that doesn't cost essentially anything. So I think it's pretty cool. Before I go, I want to just, I think I have another 5 minutes. I want to describe you quickly like this other system, because it's new. This is, I would say, is not cellulose, so they are not. We still are not able to create them on having more biocompatible, because these are like standard blockopolymers that are made out of polystyrene and PDMS block. So these are obviously not. They are not truly sustainable material, but in principle, you could try to think about substituting this block with more biocompatible molecule, and many groups are working on this. So we really hope to get more biocompatible block in the future. But they are also really interesting system, because they, similarly to the cellulose nanocrystal, they self-assemble into this layered structure, and they have the right scale to be on the wavelength of the light. And if you use the same trick of assembling them in a confined geometry, you can similarly obtain nicely colored pigments. And what I wanted to show you here is that the big advantage now, since you have a polymeric system instead of colloidal system, the processability of the method, it's really fast. So you can obtain this mixture like in really 5 minutes self-assembly time, while the cellulose, it's a little bit longer. We need a few hours in order to get the same level of intensity. And then you can get, again, color in the entire visible range that are really bright and interesting. And as you can see here, when you look at the inside of the spheres, you can again recognize all this layered structure, and the layered structure is more or less on the same scale that what I was showing you before with the cellulose, in such a way that you can produce this coloration. What is cool is that, as I was telling you before, now, since the structure, the spheres are a little bit larger, when you look at them, this effect of hierarchical structure, because they are so ordered, so perfect, you still maintain some angular dependence optical response when you look at them in direct illumination. Instead, when they are in diffuse illumination, the color again is constant independently from the angle of observation. But if you slightly change a little bit the chemistry and you adjust the blocks, again here, you can create what we call the correlated structure, where the system is slightly different, but you still have Now you don't have layer anymore, but you have semi-periodic, actually correlated system of holes inside the structure. And this one, they can actually give rise to a much more angular independent color. And when you look at them, for example, now for different angle of illumination, really wide range, again, here you can get color that are fully angular independent. And why do I insist so much with this angular independent color? I think it's really important because often when you look at the majority of the pigment that we are surrounded by, apart this special interference pigment that we have on car, on some glitter and cosmetic, we often use coloration that are angular independent, that look all the same. So the ability to create this type of structure using this interference effect is becoming more and more important, and many more groups are also working into this topic. And however, this, there is always challenges and advantages and disadvantages. When you want to work with this angular independent color, there are much more challenges to produce coloration like red. And this is like, when you go and look at the different also commercial alternatives to produce red that are like more challenging to obtain. But for example, for blue, which is one of the most desired color, also because pigments in the type of pigments that can make blue are more limited than the one that you can get, for example, for red coloration. So in the case of blue, it works really well. So depending on the type of application that you need and the type of optical response that you want, you might find the right type of structure. And the really last few minutes, if I still have time, Monica, you tell me. I think it's 37, so I should be fine.
You'll be fine.
Okay. And this is like a little bit more science. Is a little bit more, I would say, advanced. We are not yet on scale like this material that I think they are really ready for commercialization. I would just go show you some example of what I like to call bionic material. So that it's a new concept of what we are recently working more and more on, the fact that instead of using part of the living organism, like we take the cellulose from cotton or trees, and then we use it to make our photonic structure. In this case, we really exploit entire part of the organism, so using living cell in order to make our optical material. And the first example is this, what we like to call 3D printed bionic corals, where we can create hydrogels that are capable to, and 3D print them with really high resolution, that are able to host living algae. So in this case, the hydrogel essentially is providing, is functioning like a coral. That's why we call it bionic corals. It's functioning like the coral that is hosting the algae inside these polyps, the tentacles. And we have demonstrated, for example, that this type of system can be really boost the efficiency of bio-photoreactor in order to produce, for example, specific type of biomass. Or similarly, in this case, we exploit bacteria, so living colonies of bacteria that spontaneously arrange into periodic structure, and therefore they create this structural color, and then we try to exploit them as living paints. And since they are living, these bacteria, in principle, you can even genetically modify to be sensor. So I think it's I hope that with this idea is like, I would like to give you a bit what is the vision behind, and I would like you that I hope that I convinced you that it's, if we really can extract material or part of the material or even like entire organism and make using more and more concept of additional manufacturing, so sustainable industry and manufacturing, we can really obtain material that are functional and actually a little bit less, that have a less strong impact on our environment, so that also again we are helped to reduce this waste, or maybe exploit this waste for preparation of new functional nanomaterial. So with this, I hope that I entertained you enough, and I hope you enjoyed the talk, and I really would like to thank you for your attention.
Thank you, Silvia.
maybe I do. Just stop sharing.
Thank you very much. Are there any pressing questions that people may want to ask but have not yet put on the chat? I have a question. So Dan Mayhew, would you like to unmute yourself and ask the question?
Otherwise, I can
Otherwise, I can read it. You have methods to use the cellulose droplets to work with inkjet printers. Do you have methods or plan to make it work in spray coating manufacturing? Given the sustainable aspects of these pigments, it would be great to use them for the new generation of sustainable paper-based packaging, or use them to green up the current packaging. Sorry, is it noisy office? Okay, there. Dan, thank you very much.
And thank you for the question. It's a little bit more challenging because, as I was telling you before, in the case of the cellulose, the self-assembly time it's a bit longer. So in a simple spray deposition, you remove the solvent really fast, and therefore you leave a small amount of time for the self-assemble to happen. But actually, there are tricks where we are like investigating. But it makes it's a little bit more complicated in terms of the application times and the temperature condition. They are a little bit more difficult to control all at the same time. So it's not that it's impossible, it's possible. We can already spray them, but the quality of the optical response that you obtain is not as nice as what I've shown you in the results so far. So we think that actually it might be a little bit, maybe you have an extra step, but then you have the self-assembly that is performed in a droplets form in emulsion, and then you can recollect this emulsion and then spray that. But it depends a bit also on what type of printing. So in this inkjet printing, we directly print the suspension, and in this small volume with the right characteristic, you can obtain good results. But if you want to do large scale coating, really wide spray coating, then it is more complicated because you are affected, is much less local and much less control, and this lack of control provides you optical property that we still didn't manage to fully optimize.
Thank you. Another question to you, have you got experience with natural materials other than cellulose? If yes, which ones?
We have worked with chitin, and actually you can do exactly the same of what we have shown so far. It's a little bit more complicated because so the chitin that is low, essentially that is commercially available, comes from shrimps, from shrimp cell, the shrimp leftover, and that's this chitin tends to assemble in forming architecture that are larger pitch, so that they don't work well for the visible range. That's the first problem. If you get into other source of chitin, like mushroom chitin, then you can really reach the visible range. But in terms of response, the intrinsic birefringence of the chitin is not as good as the one of the cellulose. So the cellulose is actually also luckily a good material in terms of optical response, just because of the polymer backbone. So I think it's just like a kind of a lucky coincidence, I would say, because that's the intrinsic property of the material. So in this case, it's easier with the cellulose than the chitin.
Can you concentrate your hierarchical pigments, block or polymer or the cellulose, and then use them as ink in printing for angel independent color?
Yes, this is like, I show you two different examples. So both of them, when you concentrate them and then you redisperse them, this is so the covering, maybe in the slides, the covering that I show you, they were not ideal, but because we just have one, like some of them deposited. But in principle, if you put a more layer on top of it and you have a much more concentrated structure, this is what you would get an angular independent, because they are intrinsically angular independent.
You mentioned that the colors work best with an observer in the background. Are you also working on introducing absorbing properties into the structures themselves?
Yes, we are trying a little bit with some lignin and some other type of absorbent like carbon black, and you can combine it. We can also not interfere too much with the self-assembly, and this is in principle, it's something that it's easy and possible. And actually, this is what also the majority of this interference pigment that you find commercially, they always have an absorbing layer. So this is standard, and we generally, since we do it mainly for the science and not too much for application, this is just something that you add on, and then when you want to make a nice picture, you remember to take it with the right background.
Nicola is asking a long question. Nicola, would you like to ask the question?
I think it's talking about what is the reliability, how much do they last, and how much they survive,
Yes.
and what you want.
Can you protect? Yeah.
I think it's like, this is a good question, and I think it's a question that everyone asks, and then this depends on the formulation. So every pigment, also like the commercial pigment that you buy, they rely on the formulation. So if you use if you put them on top of a car, you have a finishing that covers the car, that makes the material also that it's resistant to water, resistant to solar damaging and stuff like that. So per se, cellulose, it's a good material because it has a thermal stability up to 250 degrees. So it's really stable. The color is stable because it doesn't bleach, because it's not the pigment, but it's structural color. So you don't have this problem of bleaching. Yeah. But then depend if it lasts or doesn't last, depend on the formulation. And obviously you have also to think about what is really sustainable, right? So if you want to have something that is in a cosmetic product or in a powder, that you wear it in once, then you wash your face, it ends up in the sink, you actually don't want that it lasts too long. Yes, you want that it actually can be attacked. Yes, so you want to have it as natural as possible. And I think in that case, the cellulose would be great, because we are also working with some collaborators in Angela Ruskin University that are actually testing this. What is the environmental impact of this particle once we release them? Yeah. So how long do they last when they end up in a landfill or in water waste? And we know so far that for the study that many people have done on this work, we are not the only group working on the system, on these nanomaterials, but many other people are working on that. We know that, for example, for aquatic life, they have a really low impact. So they didn't demonstrate that it was problematic for aquatic life. So I think in this case, it's a good material. If you want to put it inside a car, then you will need to work on a formulation. And obviously then, the compostability and the stability of all the system would need to be evaluated in altogether. So it's a problem, it's a little bit more complex.
Yeah. The next question, I think, sort of ties in with that. That is, what are the concerns over the human intake of microparticles of man-made plastic films? I guess it comes with what you're saying, that some need to be faster, some less.
They depends a lot, and this is also like, in terms of the human intake, if you means like eating it. Yes, when you eat them, for example, you know that some microplastic, they are not problematic. You just digest them and then they go away. This, like some of the cellulose that are food grade and compatible, they are also known that if in large amount, they can create problem with the bacteriosome in the gut, for example. And this hydroxypropyl cellulose that I show you we eat, that is in many commercial product already available. Like if you buy ice cream that is not a good Italian ice cream from a good Italian, but if you buy this ice cream that you buy like frozen ice cream, you often have this hydroxypropyl cellulose. If you buy frozen pizza, you also often have this. So I think on a really large amount, the hydroxypropyl cellulose can be problematic. About the cellulose and chitin nanocrystal, we still don't know because there is not enough data. Some studies suggest that the cellulose is not really interacting much with the microbiome that you have in the gut, because it's is not the same type of fiber that you have in the salad, that is more, that also has amorphous and also this mixed to other polysaccharides. So it seems that it's pretty less interacting, but we don't have, it's also a pretty new material. So for like ingestion, the cellulose nanocrystal, we don't know. The hydroxypropylcellulose, it's a food approved, but it's also, I know that it's like in by looking at some recent study, might give can give some problem with the microbiome.
Thank you. Do CNCs self-assemble in constrained space, for example, inside a few microns capillary?
Few microns capillary is not a problem. If you go even smaller, that you might you can create anchoring effect on the surface that might disturb or influence the helicoidal orientation of the cholesteric structure. So it depends a little bit on the type of system. But if you take a micron, few microns capillary, you will nicely form the helicoidal shape, and then the anchoring at the surface is like the crystal will be parallel to the surface, and then you can actually induce a good ordering within the cholesteric structure. So we use them to also do phase diagram.
Next question. What do you think the problem is with industry still using harmful artificial materials and no sustainable materials?
It's a general question, and it's like, it depends on the answer. So the point is that, I think at the moment, the way that we make, I'm being fully honest, the way that you make now cellulose nanocrystal is not, I wouldn't say it's the most sustainable way of making them, but at least it's a material that could be actually harvested from biomass that is unused. So in principle, I think you need to really create, and I am a believer of this circular economies where, you actually produce a specific material from the sources that you have as local as possible, and then you can exploit them for production of other things. So I don't think that it, in a place where there is abundance of this biomass, it might make sense to use this material for producing, this new colorants. If we think about then starting to cut tree in the Amazon forest in order to get the good quality of CNC, you don't solve a problem, you create another one. So this, it's a little bit, I'm sorry, I don't have the knowledge to really create a global, I think it's like itself has the potential to be, but then when it goes into industry, I think it's the responsibility also of legislation that this type of material are exploited in the right way, produced in the right way, and then trying to find the right market in a good way.
Yeah, Roberta asks whether you use sustainable chemicals to produce your fascinating cellulose-based materials.
No, we don't, because we do, the first treatment that we do is sulfuric acid hydrolysis. That's what kills you already all your nice idea of sustainability. There are enzymatic treatment that you can exploit for producing this cellulose, but the problem of yield, it becomes a little bit less, the yield is much less efficient. So there are ways to produce them more sustainably, and I think there are coming more and more. But the one that we made, that we are using in the lab, that we have optimized our recipes, if you want, they are produced with the sulfuric acid hydrolysis. Obviously, you can always recycle part of it, so you can implement a little bit greener method to do so. But starting from it is not a truly, it's not a really, you wouldn't use the green, it's not, sulfuric acid is not one of the green chemistry recommended solvent or acid to use.
Thank you. I think we have gone all the way down to the last of the questions. It leaves me to thank you very much for coming and giving us this exciting and fascinating talk. And I guess I'll ask Louise whether there's anything
Yes,
else.
thank you. Thank you, Silvia, for a fascinating talk, and to Monica for chairing so brilliantly and reading out the questions. That was fantastic. We have recorded this session, and there were quite a lot of videos within the talk, so hopefully we can process that and be able to send you the link as well. It's literally a sort of capacity thing, basically. Yeah, we'll send that round to you. We've got another talk. It's not with the Maxwell Centre, actually. On chemistry and digital molecular technologies on the 14th of January with a new centre that's opened up with the University of Cambridge. So I'll pop the link into the follow-up email for that as well. But thank you so much. That was a fascinating talk. Even me with a non-science background could understand quite a lot of it. So thank you. And thank you, Monica, as always, for rounding up a fantastic speaker, and to Aga, I can see there as well. Hello, for your support and for letting us run these on a termly basis. So yeah, have a good break if anybody is coming up for a Christmas break. We're just about to log off now. We'll be around next week, but then the week after that, we all have holidays until January. So,
thank you again. Thank you for listening, and yeah, we have a good Christmas and Happy New Year.
Yeah, brilliant. Thank you very much.
Thank you.
That's fantastic.